Thank you for visiting my poster. Microplastic pollution is a growing global concern due to its persistence, ubiquity, and potential impacts on marine organisms and human health. Microplastics, defined as plastic particles smaller than 5 millimeters, have been increasingly reported in commercial fish species worldwide, where they are ingested through foraging and trophic transfer. Our study aimed to quantify microplastic abundance, characterize their physical features, and perform FTR polymer analysis to identify and assess the hazard potential of polymers present in fish gastrointestinal tracts along the southeast coast of Chennai, Tamil Nadu. Species specific findings. Our results revealed widespread microplastic contamination across all sampled species. Among them, Thrisa mystax, from Kasimedu, Chennai showed the highest concentration of microplastics. This may be influenced by species specific feeding behavior or local environmental conditions such as proximity to fishing activities, coastal litter, and industrial discharge. Overall, the findings indicate significant anthropogenic pressure on the Chennai coastal ecosystem, affecting commercially important fishes. Microplastic characteristics, color, shape, size. When we examine microplastic characteristics, black microplastics were dominant, suggesting sources from fishing gear, nets, ropes, and industrial materials. Fibers were the most abundant, likely derived from synthetic textiles, domestic wastewater, and fishing activities. Their lightweight and flexible nature enhances bioavailability to fish. Most microplastics were 1 millimeter, indicating that smaller particles are more easily ingested, may penetrate intestinal tissues, and reflect advanced fragmentation of larger debris in the environment. Polymer identification and risk assessment (FTIR) analysis. We used FTIR spectroscopy to identify the polymers present and evaluate their hazard potential. Polymers detected included polyethylene, polypropylene, polystyrene, polyethylene terephthalate, and epoxy resin. Among these, epoxy resin had the highest hazard score. It contains bisphenol A and reactive additives which are persistent in the environment, endocrine disrupting, cytotoxic. Epoxy resin can also absorb hydrophobic pollutants, increasing ecological and human health risk. Other polymers like PE, PP, PS, and PET contribute primarily to physical stress and potential chemical exposure but are lower in hazard than epoxy resin. This analysis highlights that FTIR is crucial for both polymer identification and risk prioritization, allowing us to focus on the most hazardous polymers affecting marine life. Ecological and human health implications. The ingestion of black, fibrous, small-sized microplastics, especially by Thrisa mystax from Kasimedu, raises several concerns. Trophic transfer. MPs can move up the food chain impacting predators. Physiological impacts. Small MPs and hazardous polymers like epoxy resin can cause gut blockage, oxidative stress, and tissue damage. Seafood safety. Presence of toxic polymers in edible fish raises potential human health risk from consumption. Conclusion and recommendation. In summary, our study demonstrates high microplastic contamination in commercial fishes, with Risa mystax from Kasimedu, Chennai showing the highest ingestion. Dominance of black fibrous 1 mm sized microplastics reflecting coastal anthropogenic pollution. Presence of high risk polymers, particularly epoxy resin, with ecological and human health implications. These findings emphasize the need for improved waste management and recycling, monitoring and regulation of industrial discharges, substitution with eco friendly materials. Our study provides critical insights for mitigation strategies to protect marine ecosystems and ensure seafood safety. Acknowledgement and thanks. Finally, I would like to thank my research team, laboratory staff, and funding organization for their support. Thank you for all your attention, and I would be happy to answer any questions.